Good day, learners! Welcome back to my YouTube channel. For today's lesson, I will be discussing the first part of Module 2, which is about cell cycle. But before I proceed on my lesson, if you're new to my channel, don't forget to subscribe to update you on my latest lesson vlog. Cell cycle is important in the process of forming plants, repairing of tissue, and forming life. Our learning objective for this lesson: explain the stages of cell cycle. As new cells are produced during cell division, either mitosis and meiosis, the cell passes through different stages of development or known as cell cycle. So what is cell cycle? It is a series of events that leads the cell to duplicate their genetic contents and divide into daughter cells. The entire cycle is divided into two main stages. The first one is interphase. Interphase is divided into three stages, namely the G1 phase or gap1 phase, S phase or the synthesis phase, and G2 or gap2 phase. The other one is the M phase, where M stands for either meiosis or mitosis, where the cell undergoes division. The whole process of cell cycle are shown in your screen. It explains the beginning of heredity and variation, wherein it composed of different stages. So let us discuss the first part of the cell cycle, which is about interphase. So what happens during the interphase? During the interphase, it is known as the preparatory stage or non-dividing stage because it focuses on the cell growth and replicate its chromosome to prepare cell division. It is also a part where the cell is most active metabolically because it engages into different vital functions such as nutrition, respiration, and biosynthesis. It also spends 90% of time in the process. The interphase consists of three stages. The first one is the G1 phase or known as gap1 phase. The cell at this stage is still young and it undergoes rapid growth. In this stage, the organelles are formed and proteins are produced. It is the longest phase among the three stages in cells. In addition, the mechanism on this stage is activated to ensure that every materials are ready and complete before undergoing the next stage. The next stage is what we call as S phase or synthesis phase. The DNA doubles at this stage by the process called replication. At the end of this stage, each chromosome is made up of two sister chromatids attached at the centromere. So why is it important to duplicate the genetic materials? So that the cells that will produce will not have any abnormalities. The last stage of the interphase is known as G2 phase or gap2 phase. It is the final preparation for the cell division, wherein it assembles protein such as microtubules, hollow protein fibers that cause the chromosomes to move during cell division. Take a look of this illustration. Each stages has its boundary or checkpoint to ensure that the process of each stages are complete before entering the next stages. The purpose of this checkpoint is to ensure that all materials in each stages are developed and complete before allowing it to undergo the next stage and to avoid abnormalities in the whole cycle. Aside from G1 phase, S phase and G2 phase, there is another phase called as G0 phase or known as inactive phase. On this stage, cell cycle may stop for a specific kinds of cell. Cells in this stage are not actively preparing to divide. Some cells enter on this stage temporarily until there is an external signal or force that triggers the onset of G1. Examples of the cells that undergo G0 phase are the cells found in heart muscle, eyes, and brain. These cells are function metabolically even though they are not ready for division. That's why if these cells are damaged, they may not be able to reproduce it by themselves. After the interphase, to complete the whole cell cycle, the cell may now ready to undergo cell division. We have two kinds of cell division. The first one is mitosis. It is a type of cell division where multicellular organisms undergo growth and development and when they repair damaged tissues. The other one is meiosis. It is a type of cell division used to produce sex cells such as egg and sperm cells. When cell divides, technically, nucleus and cytoplasm also divides. After the division of the nucleus or known as karyokinesis, the cytoplasm starts to divide, known as cytokinesis. Cytokinesis in animal cell and other cells that lack cell walls starts at the exterior of the cell moving inwards until the cell is divided into two cells. This process is called cleavage furrow formation. In plant cell, the cell divides, forming a cell plate on its center that is eventually become a cell wall described as a protective layer of plant cell. This is how cleavage furrow formed, as I mentioned earlier. To check your understanding, it's quiz time! Thank you, grade 8 students. I hope you learned something. If you liked my video, don't forget to like, share, subscribe, and turn on the bell for more updates in our science class.